All right, so as advertised, I'm Lauren Egley, and I'm a PhD student at UConn. Going to talk a little bit about some of my work evaluating ground beetle recovery post-harvesting Quebec's boreal mixedwoods. So we'll start with a little bit of context to frame out my study. So as we know, managed boreal forests are harvested mostly through clear cutting. And when clear cut stands of any successional stage typically reinitiate as even-aged hardwood-dominated stands. And when a forest is fully regulated through that kind of even-aged management, the area typically won't have any stands which are over the rotation age. So for example, in this figure, it's a rotation age of 100 years, so all of our stands are 100 years or younger, because of course, as soon as they hit that maximum age, they're cut again. In contrast, when you consider the impacts of a stand replacing natural disturbance, like a forest fire, you get a completely different distribution of age classes. And that's because the probability of burning is independent from the stand age. So a 100-year fire cycle, instead you get a distribution where a third of the stands in that area are actually older than 100 years. And those older age stands can be important because they provide greater structural variation, more diversity in tree species, microhabitats, and all that sort of thing is necessary for some species to be able to thrive in that area. So therefore, some forest management operations have attempted to integrate a greater variety of harvesting techniques in the hope that they'll be able to imitate this wider distribution of stand ages and classes and represent something that's more similar to an unmanaged area. So one of these strategies is the cohort model. In the cohort model, partial cutting can be used to emulate competition by thinning young trees or emulate senescence by harvesting larger stems. And this can move a stand from that first cohort, which in our case is hardwood dominated, to the second cohort, mixed wood dominated, or vice versa. You could also conduct single tree selection or group selection in the second cohort stands to move them towards the third cohort, which in our case is softwood dominated, or vice versa. Can cut those same types of cuts in softwood stands to keep them softwood, as long as you keep your gap sizes relatively small. And then of course, finally, you could clear cut any cohort and it should bring it back to that first hardwood dominated initiating cohort. So my goal for this study was to examine the effectiveness of that kind of cohort model for preserving biodiversity, and if biodiversity was preserved, determine how long it took for resident ground beetle communities to recover following that type of harvesting. And I picked ground beetles to use in this study because they're a diverse taxa. They readily respond to changes in forest composition and structure, and then they're also really highly sensitive to stand level disturbances. So my study site is in the Lake du Parquet Research and Teaching Forest in Quebec, and my project took place as part of the SAFE project. SAFE was initiated in the 1990s and cuts happened within hardwood, mixedwood, and softwood dominated stands. In the hardwood stands, we had a 1/3 cut which targeted small stems, a 2/3 which targeted bigger marketable stems. And then we also had clear cuts where the residues were further treated through prescribed burning, were removed as part of poultry harvest, or were left on site. And then we had controls that were left uncut. In the mixed wood stands, we had harvest through clear cutting, then also through partial cutting in the form of single tree selection that was 40% of the basal area, or group selection, also 40% of the basal area, but this time in 400-meter square islands. And then again, uncut controls. And then finally, the softwoods were either clear cut or left as controls. So in those areas, we collected ground beetles from 360 pitfall traps, and that occurred over 7 sampling seasons between 2004 and 2018. And during that time, we collected and identified 71,000 carabids from 74 species. So on to my results. So this is ground beetle community response, shown as a sum of squares multivariate regression tree, or SSMRT. And I picked an MRT for this analysis because MRTs directly relate that species response to our explanatory variables, and they don't make a lot of assumptions. So for example, it doesn't assume relationships are linear like an ordination would. And it also gives us some pretty definitive results that are easier to interpret. And that was important for this analysis because we're looking at the interactions between time since harvest, harvest treatment, and cohort type on ground beetle community composition. So this MRT organizes the stands based on the similarity of the communities. found within each treatment. So if it regroups a harvested treatment with an uncut control, we know that that community composition is now similar, so we can consider those stands recovered. So I want to talk about this MRT, break it down a little bit into 3 segments. That far left segment, that's the earliest harvest data, so that's 3 to 10 years post-harvest. The middle, that's about 15 years post-harvest, and then that far right is our latest data, that's 20 years post-harvest. So to start with the earliest data, here we can see that right after harvest, the clear cuts in the softwood stands, the hardwood stands, the prescribed burns, and the whole tree harvest all had an immediate negative impact on ground beetle communities. In that same time frame, though, the partial cuts in the hardwood stands, so that was the 1/3 of small stems and the 2/3 of those marketable stems, those actually maintained their ground beetle communities the same as the uncut control. So that was pretty cool because it indicated that even up to that 2/3 cut was enough to preserve those resident ground beetles. However, in the mixed wood stands, where we also did partial cuts, in that case it was a 40% single tree selection or 40% group selection, did not have the same early retention of biodiversity. In those stands, we had a grouping between the clear cuts and the partial cuts. So those were similar compositions and didn't regroup with their control. So we know that those didn't maintain their communities the same way early like the hardwood stands did. So onto the middle of the tree. As of 15 years post-harvest, we still had those changes in the prescribed burn and the whole tree harvest. Those hadn't recovered yet. However, in contrast, the clear cuts in the softwood stands and the hardwood stands had recovered by 15 years. So we know that because it regrouped with their controls. The partial stands, or the partial cuts in the mixed wood stands, we also had temporary signs of recovery, but as you'll see in my next slide, that recovery didn't actually persist. So as of 20 years post-harvest, those prescribed burns and whole tree harvests, those finally recovered. But those mixed wood stands, like I said, the partial cuts and the clear cuts diverged from the uncut stands again, so we didn't get that same persistent recovery as we did in the hardwood and softwood stands. So the MRT looked at trends in community level data. Now these next models look at the trends in the top 8 most abundant species individually. So for these species, we used generalized linear mixed models, and we compared the relationship between changes in abundance with changes in basal area, time since harvest, and cohort type. And then we also included individual stands as a random effect. And we found that for half of our core 8 species, abundance increased with increasing basal area, and all 8 species increased with abundance over time. And not only did those 8 species increase with abundance over time, but also overall abundance in ground beetles as a whole increased over time, regardless of silvicultural treatment in all of our stands. So on to a few discussion points and conclusions. To start with, that significant overall increase in ground beetle abundance across all of our treatments is in marked contrast with a lot of recently reported declines in insect abundance elsewhere. I'm sure you guys have all seen articles saying like the insect apocalypse is upon us. And why we're seeing this substantial increase, while it feels like everybody else is seeing a decrease, is a bit of a mystery for us. But one possible explanation could be that the initial effects of increasing temperatures in our region are being offset by ample precipitation and an earlier snowmelt. And that combination of temperature increase by just a few degrees plus plenty of moisture, maybe putting our area's boreal forest into like a sweet spot where there's a short-term increase in productivity. And that idea was coined as the Boreal Refuge Hypothesis by d'Orangeville in 2016. And some studies have shown that increased forest productivity can translate into increased ground beetle populations. So that's one potential explanation for why our insect abundance is going up. It does seem likely that ground beetle recovery is tied to the retention or reestablishment of stand structure and overstory cover. So we saw this in the ground beetle community response, since that 1/3 and 2/3 partial cut in the hardwood stands retained beetle communities right after harvest. And we think that was likely because those stands had a significant number of green trees left behind that kept a canopy for them. Similarly, in the clear cuts, By the time the beetle communities began to recover, which was 17 years post-harvest, by that point, the aspen stems were an average of 10 centimeters in diameter and over 3 meters tall. So again, we had that recovery of an overstory kind of paired up with the beetles recovering. So overall, that's promising for a coarse filter approach to preserving their biodiversity. So if you can maintain or reestablish that canopy cover relatively quickly, then you can likely maintain or facilitate the recovery of those ground beetles following harvest. Then we also found that while clear cutting reduced the initial abundance of the ground beetles, those additional manipulations of the logging residues, so that was the whole tree harvests and the prescribed burns, those delayed recovery even further. And for some of the less common species, which already had a negative response to overstory removal, those successive disturbances had an additive impact and further depressed the population. So it had a pretty big impact on those rarer species. So therefore, we might want to think twice about policies which promote that additional removal of biomass, since there was pretty significant negative impacts on biodiversity. And then finally, although recovery in terms of maintaining the resident species was a step in the right direction, we didn't find much evidence that that cohort model could actually be used to control the transition of fauna between the different successional stages. So as a reminder, in the hard and softwood stands, the beetles recovered to resemble the communities found in their uncut controls from their original cohort. So we expected them to shift to a different cohort, but instead they went back to what they would have been had they not been harvested. And only in those mixed wood stands did we see the possibility that the harvest might be transitioning towards something else. So those ones recovered briefly, then diverged again. And one explanation for that divergence could be that those communities are starting to their transition to a different cohort. But as of 20 years post-harvest, we don't have a clear trend on that yet. So we'll have to wait and see what happens in the coming years. And a thanks to everybody who came before me and set up the project, and ready for questions if there are any, or there's also, I have my email up there if anybody wants to chat further as well.
As Tim introduced me, my name is Ursula Dergovsky, and I'm going to talk about the long-term effects of high and low intensity partial cutting on saprophilic beetle communities in aspen-dominated stands of the Eastern Boreal mixed-wood forest. I work with Tim, who is my director at UCAM University, and Miguel Montoro Girona, who is my co-director at UCAT University. So, to begin with, I will talk about natural disturbance-based management. Now, as the name implies, in natural disturbance-based management, we aim to create a level of disturbance with our harvesting that the forest would naturally experience through events such as wind throw, fire, or insect outbreak. And one way we do this is through partial cutting. So in partial cutting, we go into a stand, we remove some of the trees, but we leave some behind with the implication that over time, these trees will grow, mature, and die, presumably providing fresh deadwood into the stand maybe decades after harvest. And this is beneficial because much of the biodiversity in a stand is saprophilic or dependent on deadwood. Now, if I wanted to go and study how saprophilic biodiversity recovers long-term after harvesting, which organism should I choose? Well, why not choose beetles? Because beetles are not only a very diverse group of organisms, they're also very numerous. So 1 in every 4 animals is a beetle, and they also occupy many different feeding guilds, as well as environmental niches. So if I have beetles as the representatives of saprophilic biodiversity in my study, I'm getting a representation of how many different environmental niches are affected long-term after harvest. So the question that I was interested in answering is 20 years after a low intensity partial cut where we remove 1/3 of trees from a stand and a high intensity partial cut where we remove 2/3 of trees from a stand, have saprophilic beetle communities recovered? So my hypothesis was that the community composition, the overall saprophilic beetle abundance, and the richness of saprophilic beetles will not be significantly different between the control sites and the sites where we have 1/3 partial cuts. However, in contrast, I believe that in the 2/3 partial cuts, these parameters, community composition, overall saprophilic beetle abundance, and the species richness will be significantly different in two-third cuts. And in fact, the two-third cuts will more closely resemble clear cuts. So where should I go test this hypothesis? Well, over 20 years ago in the Sylviculture et Aménagement Forestier Écosystémique study, or the SAFE study, in the Lac du Parquet Abitibi-Témiscamingue region of Quebec, is specifically in the hardwood stands. In the winter of 1998 to 1999, we had a 1/3 partial cut done in which we removed small non-vigorous stems in a low thinning technique. We had a 2/3 partial cut where we targeted large diameter marketable stems in crown thinning. And we had two types of clear cut. So one with a prescribed burn and one without. And CPRS just refers to the type of clear cutting commonly used in Quebec, in which we allow for some understory protection. We came back May to August 2019, so over 20 years later, and we set up IBL traps in the stand. So we had 3 blocks. In each of the blocks, we had the treatments, and in each treatment, we installed 3 traps. So total, we had 9 IBL traps in each treatment. We had 3 collections between May to August of 2019, and the beetles were identified, and then we classified them as saprophilic or non-saprophilic. And of course, in this study, I'm only focusing on the saprophilic beetles. So let me show you some of the results from this. I will say that all of the results I'm showing you are based on 5,902 individual beetles that I ID'd and I classified as saprophilic, and these belong to 301 different species. So first, let's look at the species richness through this rarefaction curve that I have over here. As you can see, 20 years later, it is both of the clear cuts with and without a burn that have a lower species richness than either of the partial cuts or the uncut control stand. So we don't see the recovery in species richness 20 years later in the clear cuts. However, in the partial cuts, the species richness is similar to the uncut control stands. Now, what about our other parameters? So first, let me start with community composition. I will not be showing you an MRT tree because after analysis, what I found is actually community composition between the treatments did not differ. Now, where there was a difference was in the overall abundance of saprophilic beetles 20 years later. I want to start by saying that all of the treatments, whether it's a partial cut or a clear cut, all of them differed significantly in the abundance of saprophilic beetles 20 years later. However, if you look at this graph, you see that in the 1/3 partial cut, there's a much higher abundance of saprophilic beetles 20 years later than in the 2/3 partial cut or the clear cuts. In fact, you see in the 2/3 partial cuts, there's not really any benefit to this type of partial cut over a clear cut, whereas the 1/3 partial cut is more promoting that recovery in the abundance. Now, let's make this more interesting and break it down by the feeding gills. So I have for you here abundances of fungivores, xylophages, and predaceous beetles. Let's look first at the middle graph, where I talk about the fungivorous beetles. So what you see is a similar pattern to the overall saprophilic beetle abundance when it comes to that 1/3 partial cut, offering more of that benefit to the recovery of the abundance 20 years later. Now, if you look at the graph of xylophagous beetles on the right, you still somewhat see a bit of this pattern, but what I want you to focus on in this graph is that it seems like it's the clear cuts that have that reduction of xylophagous beetle abundance 20 years later. And the graph on the right with the predaceous beetles, we start losing these patterns that we're seeing in the other guilds and overall saprophilic beetles. So what we can infer from these graphs is it seems like it's mainly the fungivorous beetles that are driving this pattern of an overall saprophilic beetle abundance benefiting from 1/3 partial cuts. So we can ask ourselves, what is influencing the fungivorous beetles and on a larger scale, the overall saprophilic beetle community, and that abundance to be more higher in the 1/3 cuts 20 years later? Well, we are studying saprophilic beetles, so it's useful to now look at what does deadwood look like 20 years later, which I will do right now. So you see on the left top graph, again, I have for you, I'm repeating the graph from earlier with the overall saprosillic abundance 20 years later. You see that it's highest in the 1/3 partial cuts. However, if we look at the volume of deadwood 20 years later beside it, we see that actually the highest volume of deadwood is in the 2/3 cuts. Now, there's different explanations for this, but one of which can be if we go into a stand and we harvest 2/3 of trees, we leave behind only 1/3. This 1/3 is a lot more vulnerable to mortality from wind throw. So maybe we went in, we took all these trees, the 1/3 that was left died in the 2 decades from the wind throw. So now we have all this deadwood in the 2/3 partial cuts. But then why don't we have more beetles in 2/3 partial cuts instead of 1/3? Well, as you recall, it was the fungivores that were mainly influencing this pattern of the 1/3 really being beneficial. So we might ask ourselves, maybe it's not the deadwood but on its own that's influencing recovery of saprosillic beetle abundance. Maybe it's deadwood that is colonized by fungi, because saprosillic fungi are very important in the saprosillic community. They often make the deadwood more habitable for other saprosillic organisms. So I think it would be very interesting if someone went and did a study similar to the one I'm doing, but on recovery of saprosillic fungi 20 years later. And maybe we would see a connection between the volume of deadwood, abundance of saprosillic fungi, and abundance of saprosillic beetles. Now, I'm coming back to the Xylophagous beetles graph on the bottom right. As you recall, I said that in this graph, it was the 2 clear cuts that had a reduction in the abundance of Xylophagous beetles. And I find it interesting now, if we look at the volume of deadwood by decomposition class 20 years later, we see that it is the 2 clear cuts that are have a reduction in the fresh deadwood. So this is interesting because it might explain why we see that reduction of Xylophagous beetles, which are often very dependent on fresh deadwood. Okay, so what do I want to say to conclude my study? So first, again, I want to emphasize in all of the cutting treatments, whether it's a low intensity partial cut or a high intensity partial cut, in all of the treatments and the clear cuts, there is not recovery of, there's not full recovery in these saprosillic beetle communities. This means we need longer than 20 years to see this recovery. However, as we saw, 1/3 partial cuts did offer a benefit. There was a much higher saprosillic beetle abundance 20 years later in 1/3 partial cuts, whereas in the 2/3 partial cuts, we actually did not see an advantage for recovery in terms of saprosillic beetle abundance. Now, importantly, we also see that it's not just deadwood that is affecting saprosillic beetle community recovery. We have to consider other factors. So it's possible maybe the recovery of saprosillic fungal communities will equal the recovery of saprosillic beetle communities. Okay, thank you everyone for listening to me. I really enjoyed speaking to everyone today. And if anyone has any questions, I'd be more than happy to try answer them.
There you go.
Yeah. So, hello everyone. My name is Akif Hassan and I'm a student from UCAT's Amos campus, and I'm going to present about the unique biodiversity of Esker Lakes. And this project has been supervised by Professor Miguel, Professor Guillaume, and Professor Louis Ambel. First, I want you to imagine our world like this. I know it's hard because we are experiencing global warming, but it was the situation of our world during the last ice age, which ended about 10,000 years ago. During that time, 97% of Canada was covered in thick layer of ice. The thickness of ice was up to 2 kilometers in several places. During that time, a complex geological formation was created that now we call esker. Eskers are made of a linear accumulation of layers of sand and gravel. Now, these eskers are distributed over all northern countries, including Canada. Here you can see all the eskers from Canada, and on the right side you can see the St. Matthew Lake Berry esker from Emerse Abitibi. Lakes on eskers are connected with groundwater system, and they are not connected with other river system. And this type of system actually makes them as a closed basin wetland. And this type of relationship also affect the water temperature, water quality, and nutrient content of the lake. Also, esker provides several ecological and economic services. For example, they provide sand and gravel, which is used for constructional purposes. Eskers also provide the best quality fresh water, and which is recognized as one of the best fresh water in the world. Also, esker provide opportunity for microtourism because we can find very important fungi species in there. Also, esker provide the archaeological sites. For example, the eskers provide the regional identity of Abitibi-Temiskaming. Also, esker provide the opportunity for recreational sites, recreational activities. Also, esker provide the productive jack pine timber. From these services, esker fed overexploitation of resources, and it actually altered the natural bedrock of eskers. Also, forest harvesting has negative effect over biodiversity and ecosystem in eskers. Other anthropogenic disturbance such as camping, interaction also has negative effect over the biodiversity. And there is a huge knowledge gap about the biodiversity and functioning of esker ecosystem. And to eliminate that knowledge gap, we are implementing a unique food web approach. In esker lakes, waterbirds act as a top predator, but they are just the tip of the iceberg because their population depends on macroinvertebrate community and fish community. Therefore, our objective of this study is to evaluate the waterbird biodiversity associated to esker lakes and also identify environmental driver with a unique food web approach. Our first specific objective is to assess the aquatic resources for waterbird in lakes, such as type of habitat, quality and quantity of macrophyte, fish and macrophyte communities. Our second specific objective is to assess the species richness, evenness, diversity of waterbird, and record the occurrence of indicatory species which can be linked with fishless lakes on eskers. Here is my conceptual framework. You can clearly see these two types of lakes are very different than each other. In esker lakes, they usually have low water temperature because they get their water reaches from the groundwater. Therefore, they usually have high dissolved oxygen. They also have no or less number of big fish because they are not connected with other river systems. Also, they get more light because of the transparent water. Therefore, they should have more aquatic macrophyte and algal communities. And all those factors positively affect the aquatic invertebrate, which should be higher in esker lakes. Therefore, we're expecting in esker lake, there should be a rich and diverse waterbird community. On the other hand, in clay lakes, there is less light because the water are not clear. Therefore, there should be less algal communities and aquatic macrophyte. And also there is many fish because they are connected with the river system and the wetland. And the water temperature is higher. Therefore, there is less dissolved oxygen. So we're expecting there should be less diverse aquatic invertebrate. So we are expecting in clay lakes, there should be less abundant and less diverse waterbird community. Therefore, our hypothesis of this study is that the abundance and diversity of macroinvertebrate will be higher in esker lakes compared to the lakes and clay because of high availability of resources and reduced fish predation. Our second hypothesis is that the richness, diversity, and abundance of waterbird will be higher in kettle lakes on esker compared to the lakes on clay because of more availability of their food resources in kettle lakes. Now I'm going to discuss the method. So at the beginning, we had 80 lakes around this territory, and among those 80 lakes, depending on the size of the lake, location of the lake, and logistic, we selected 50 lakes. You can see all the 50 lakes from this territory, and among these 50 lakes, 25 lakes situated on eskers and 25 lakes situated on the clay belt. For waterbird survey, we use point count and flash count method to observe when two position of the lake, and they count the bird for 20 minutes. Then they walk across the lake to detect, to maximize the detection probability. There are 2 visits per lake. For fish survey, we set 3 traps per lake and we kept the trap for 24 hours. Then we identified each fish species. Then we measured total length, body weight, and space abundance. For factory invertebrate, we collected 150 sample with a different net from the 50 lakes, and then we kept the sample in ethanol, and then we sorted each macroinvertebrate sample and identified in the laboratory. To analyze our data, we use Channel Diversity Index with package vegan to compare the diversity between 2 types of lakes. We also use principal component analysis to test relationship between biodiversity and physical quality of the lakes. We also use general linear mixed model with Poisson distribution to test the effect of physiochemical variables over species richness. We also use multivariate pattern analysis to indicate species to test the association of species with lake depths. Now I will show the results. For water biodiversity, we saw that the mean channel diversity index value is lower in Escarl lakes compared to lakes on clay. Mean abundance, total abundance, richness was also low in Escarl lake. For fish diversity, we can see mean channel diversity index value is significantly lower in Escarl lake compared to the lakes on clay. Almost half of the Escarl lakes were completely fishless. However, the total richness was higher in Escarl lake. For macroinvertebrate diversity, we didn't find any significant difference. However, mean channel diversity value is slightly lower in Escarl lake. For physiochemical variables such as nutrient and dissolved organic carbon, the concentration was lower in Escarl lake. However, dissolved oxygen was higher in Escarl lake. For salinity, conductivity, and macrophyte cover percentage, the percentage was higher in Escarl lake compared to the lakes on clay. Here is the effect of physical variables on water body richness with our GLM function. We found that significant positive effect of phosphorus and dissolved organic carbon over the water body richness, and we found the significant negative effect of lake area and harvesting distance over the water body richness. For indicator water bed, we found that there is a strong association of common goldeneye and Canada goose with Escarl lakes. And in clay lakes, we found that there is significant association of ring-necked duck and hooded merganser with clay lakes. For fish species, we found only Eoloper's strong association with Escarl lakes because half of the Escarl lakes were completely fishless. And for indicator fish in clay lakes, we found the strong association of northern red pileates, northern fine scales, and fatted minnow strong association with the clay lakes. For indicator macroinvertebrate, we found the strong association of palliative family and order with Escarl lake, and for clay lake we found the Coleoptera order and giant waterbug strong association with the clay lakes. Now I have to discuss the result. So we saw that nutrient and dissolved organic carbon was lower in Escarl lake, which can be explained by the fact that Escarl lakes get less nutrient input from the watershed because of their isolation and their situation at the higher elevation in the land use. Also, we found that dissolved oxygen was higher in Escarl lake, and this can be explained by the fact that Escarl lake usually get their water reaches from the groundwater, therefore they have low water temperature. Also, we saw less macrophyte cover in Escarl lakes, which can be explained by the fact that they get less nutrient input, therefore less nutrient for macrophyte to thrive in that ecosystem. For waterbird, we saw that the significant association of common goldeneye with this ecosystem, because common goldeneye prefer fishless Escarl lake, especially during their breeding season, because they don't like competition while they breed their ducklings. For fish, we saw that there's a significant difference in Escarl lakes. The mean channel diversity value is significantly lower because almost half of the Escarl lakes are completely fishless, and the other lakes had less number of fish. Isolation of Escarl lakes and their lower nutrient content can explain this phenomenon. For macroinvertebrate, we found the significant association of stoneflies, which can be explained by the fact that stoneflies require higher dissolved oxygen in the water to survive. Also, we found that Odonata order species had significant association with this ecosystem because they act as a predator in this type of ecosystem. We saw from our modeling that harvesting activities significantly had negative effect over biodiversity. Also, several other anthropogenic activity such as mining, species production, pollution, camping, which also can alter the esker habitat for biodiversity. And lake area and lake perimeter strongly affect the waterbird habitat selection also. So to conclude, we can say that the diversity of esker lakes is lower in all trophic level of the food web. However, few and important communities showed strong association with esker lakes because they need this special ecosystem to survive. Anthropogenic activity might alter this pristine esker ecosystem. Thank you everyone for listening.
I'm Allison Oakes. I'm a PhD student at Purdue University. And as was said, I'm speaking of the response of terrestrial salamanders to the decade following timber harvest in hardwood forests. Yeah, so for some background, terrestrial salamanders are very key components of forest ecosystems. They operate in middle positions on the food web, so they eat a lot of things that are smaller than them that nothing else would bother to eat, and are eaten by a lot of larger things that wouldn't eat anything smaller, thereby moving energy up the food chain. They're also quite useful as indicators of forest health, since they are commonly found on the forest floor, but are also highly reactive to changes in the forest environment, such as toxins, but also things like increased temperatures and drier conditions, which can be strongly affected by things like timber harvesting. With the loss of the canopy, you get these warmer and drier conditions that salamanders do not like and therefore respond to. So there has been some previous research on this topic. Mostly it's been found that clear cuts have negative effects on terrestrial salamanders. Other forms of management like shelterwoods have more variable results. Shelterwoods are staged harvests, and it depends on how much canopy is retained at each stage and which harvest stage is being studied. Uneven age management like group selection and patch cuts have also shown more variable results. Some have negative effects, some have no effects. There's concerns with some of these studies that since uneven age management requires multiple entries to a forest stand and generally cover a larger area of the landscape, there may be effects on salamanders that are not captured by studies that just look at the harvest area. It's also been suggested that strategies like retaining coarse woody debris after harvest may be able to provide shelter for salamanders, since they use these structures to take refuge from warm and dry conditions. There's also concerns that this may be an ecological trap, keeping salamanders in areas they are not suited to. Notably, a lot of studies so far have only covered short-term responses to harvest, maybe up to 5 years post-harvest. In shelterwoods especially, it is heavily biased towards studying just the first stage of shelterwood harvest, with not a lot of studies on later stages. So my study sought to close some of these gaps by examining the effects of clear cuts, shelterwoods, and patch cuts on terrestrial salamanders in southern Indiana, up to a decade post-harvest. This work is extending that done by McNeil and Williams that was published in 2014, who looked at the salamander responses up to 3 years post-harvest at this same site. We expected that our clear cuts and our larger patch cuts at least would strongly and negatively affect salamanders. So this study took place at the Hardwood Ecosystem Experiment, which is a long-term study on the effects of forest management. It is planned to be a 100-year project. I believe it is in its second decade, which is very impressive. It consists of 9 management units, 3 of which are receiving even-aged management, 3 of which are receiving uneven aged management consisting of patch cuts of 3 different sizes, and 3 are receiving no management or are controls. The first timber harvest went in the fall of 2008 for all of the different types of harvesting, and the second stage of our shelterwood harvest was in the fall of 2015. Our shelterwoods are a 3-stage shelterwood harvest, so stage 1 and stage 2 have now gone in, and stage 3 will take place within the next few years. So to collect data on salamanders, we used coverboard surveys. They're blocks of wood we set onto the forest floor. We had 30 of these boards per grid, 24 of these grids in control areas, 12 in each of the even-aged harvests, and 6 in each of the uneven-aged harvests. We checked these starting before the harvest went in. They were checked every 2 weeks during our fall season and our spring season, up until quite recently. I believe 2019 was our most recent season. So to check underneath, we just lift up the boards, we look underneath, and we count the number of salamanders we find and what species. So for our analyses, we used R for statistical computing. We looked at the total captures per each field season and modeled these using negative binomial models with mixed random effects of grid and year. We also used a before-after control impact design to better compare salamander captures after harvest to before harvest and compare that to our controls as well. Because of the limitations on the backing design, we split our data into time periods of 0 to 3 years, 4 to 6 years, and 7 to 11 years post-harvest, and also modeled shelterwoods separately due to the different harvest schedule. We also collected and analyzed some environmental data. This data was actually collected for a separate project on small mammals that occurred nearby. This included herbaceous and woody percent cover, the length of coarse woody debris, and leaf litter in each of the harvest areas. These were modeled using linear mixed effects models with that same backy design as we used on the salamanders. So we could see what was happening both with salamanders and at the same time with our environmental data. For our results, the first 3 years following harvest, we saw no effects of harvest on salamanders, which is interesting considering previous research. We saw negative effects of harvest on salamanders in the 4 to 6 year period in clear cuts and patch cuts. And this continued into the 7 to 11 year post-harvest period with no signs of recovery. There was no significant effects of shelterwoods on salamanders, which was also interesting to see, either the first or second harvest. So one thing to note is that there was a drought that took place during the 4 to 6 year period following harvest. That same section where we saw negative effects on salamanders, where we saw our salamander captures decline. So that suggests that these hotter and drier periods during the drought were worse, or at least they were not able to adjust to it in areas without canopy cover to help protect the microclimate. This indicates a compound disturbance of both drought and timber harvest, which could be a very serious concern for timber management, since we are expecting droughts to become more common in this region with changing climates. It is notable that we didn't see any effects on timber harvest on salamanders in our shelterwoods, even during the period of drought, which suggests that the canopy cover that was retained through the first and second harvest in our shelterwoods was enough to protect salamanders from these effects. Notably, this study only extended 4 years past the second harvest, so if we're looking at a lagged effect on salamanders, we wouldn't have been able to see that in this study. The third harvest will be going in the next few years, and we're all very excited to see what happens when the rest of that overstory is removed. Looking at our environment, Herbaceous and woody cover both went up following timber harvest and remained high. Woody cover returned to control levels during the 7 to 11 years post-harvest period. Forest woody debris also went up following timber harvest, but it returned to control levels in the 4 to 6 year period and stayed low. Leaf litter dropped following harvest and stayed low through the 7 to 11 year period. So this could explain some of what we saw with the salamanders. It was very interesting that we didn't have effects on salamanders in the 0 to 3 years post-harvest, but as we saw with the environment, we had very high site productivity and fast revegetation with that herbaceous and woody cover going up following harvest. This may have been able to protect the microclimate by providing some shade and keeping things moist. The coarse woody debris also went up, which may have provided additional refuges for salamanders, although it's also notable that it returned to control levels in the 4 to 6 year period, which is also when salamander captures fell. So in conclusion, we found fewer and smaller effects of harvest on terrestrial salamanders than many previous studies. Overstory retention as through shelter woods may be able to protect salamanders from both the effects of harvest and the compound effects of drought. That being said, our clear cuts and patch cuts both had effects on salamanders, which may have been driven by lower amounts of coarse woody debris or the compound effects of drought during that 4 to 6 year period, or a combination of the two. So this study has found evidence of this compound effects of drought and harvest on salamanders, and considering increasing global temperatures, understanding how these kinds of mega disturbances will affect salamanders is going to be very important in the future. I'd like to acknowledge the traditional homelands of the Indigenous people upon which Purdue University is built, and thank everyone who helped or supported this project. Yeah, I believe I have time for questions.
My name is Graham Frank. I'm a PhD student at Oregon State University, working with Meg Krawchuk. And my dissertation work focuses on the degree to which young plantations and post-fire salvage logging can support the same sorts of biodiversity that we expect to find in complex early cereal forests that develop after stand replacing wildfire in the Pacific Northwest. And today I'll be focusing in particular on the bird component of this study, though we're also looking at ground beetles, pollinators, and plants as part of the larger project. So in the past, I think 20 years or so, there's been increasing recognition that complex early cereal forests that develop after stand replacing disturbance are really important for supporting a certain swath of biodiversity that we should be focusing, in addition to how those forest species are recovering after disturbance, how some of these more disturbance dependent species are doing in immediately after disturbance. And these young forests are particularly important in landscapes that are dominated by closed canopy forests, largely because there's so much light reaching the forest floor, and that can support herbaceous plants, a lot of broadleaf shrubs and trees, and the species that rely on those. These complex early cereal forests also supply abundant deadwood in the form of snags if it's wildfire or insect outbreaks, or downwood following wind throw, and the species that rely on deadwood. And in coniferous dominated forest landscapes, they can be a really important source of broadleaf vegetation that often gets crowded out by coniferous species, which are often less palatable to herbivorous animals, insects, and things that predate upon them. And when we talk about complex early cereal forests, we're talking about complexity, both in terms of the vegetation structure and in terms of all of this deadwood in the ecosystem. The composition, here you can see this tight intermixing of herbaceous plant species, resprouting hardwoods, shrubs, regenerating conifers, and then a lot of spatial complexity because of the importance of biological legacies, that determine habitat characteristics in these stands, and temporal complexity because of the rapid change that takes place in the years after disturbance. On kind of the opposite end of this spectrum, we have these intensively managed forage plantations that are really important for meeting global timber demand, but are much more simplified, both in terms of their composition and structure, in large part because they really limit the impact of these biological legacies by removing any deadwood, really limiting the broadleaf component here with the use of herbicides. And all this changes the pace of stand development, both on the front end with herbicide use and on the back end with much more rapid canopy closure of conifers, kind of shading out those herbaceous and broadleaf plants. Post-fire salvage logging is another important disturbance in this system and a lot of fire-prone forest systems. Of course, it's important for recouping economic losses after fire and can improve safety for tree planters where reforestation is an important goal. But the snag removal reduces foraging and nesting habitat for species that rely on snags and can affect microclimate. And that compound disturbance of logging immediately after fire can potentially hinder vegetation recovery or change the trajectory of the plant community after fire. At the same time, a lot of these disturbance adapted species that might rely on early sterile habitat also have a pretty broad ecological amplitude. And so our guiding research question here is, to what degree do these managed forms of early sterile forests support the biodiversity that's associated with their kind of closest natural counterpart, which in the Pacific Northwest and in a lot of forest systems is stand replacing wildfire. In addition, we're interested in how biodiversity patterns change with time since disturbance and how these comparisons differ among specific functional guilds or with functional traits. And so within Oregon, we're specifically focusing on the Klamath Mountains ecoregion down in Southwest Oregon here. And we've chosen this landscape because of this tight intermixing of public and private land ownership in this checkerboard pattern that you see, as well as several large wildfires that have burned across this landscape in the past 20 years, of which these are really only a subset that we're sampling on. And we've established and sampled at 210 field plots across, nested within 70 study sites over the past few years. Anything you see within a fire footprint here is on extensively managed public federal land. Anything outside of a fire footprint is on intensively managed industrial timberland. And so this gives us a really nice chrono sequence following intensive management from 2 to 20 years. These are clear cuts replanted with Douglas fir, herbicided multiple times, very similar to what Matt Betts was describing as this moderate treatment in the Oregon Coast Range earlier this morning. Our stand replacing wildfire sites have at least 75%, usually much greater mortality. They're at least 10 acres and have no disturbance in the last 45 years before fire, to try to match as close as we can that rotation age for the intensively managed plantations. And these are all within the same Douglas fir forest type to try to keep things as apples to apples as possible. Our post-fire salvage sites have kind of the same criteria as stand replacing fire sites, but they've experienced clear-cut style salvage logging following the fire and were replanted with mixed conifer species, but did not receive any herbicide treatments. And due to sort of limited availability of salvage logging that fit these criteria on this landscape, we just have this kind of narrower age range for our salvage log stands. So a couple of hypotheses here. Because we expect that management homogenizes early sterile forest habitat, we're expecting lower species richness in our plantations and our salvage stands. And we're particularly interested here in cavity nesting bird species because of this loss of snags that you get after fire, and also leaf gleaning insectivores, because those seem to respond really strongly to the pace of development of woody vegetation and the type of woody vegetation that's there, whether it's broadleaf or coniferous. We also expect that there are going to be really different trajectories in terms of the development of these assemblages over time. And in particular, based on some work that's been done in Canada's boreal forest, we expect to see that the strongest differences between disturbance types are at the most recently disturbed sites, and that bird assemblages will kind of converge over time as the snags fracture and fall after fire, cavity nesting species become less important, and vegetation develops in those harvested sites. So we've conducted breeding season point counts for birds at 3 points within each of our sites, visiting each of those 3 times during the breeding season. And this allowed us to fit a multi-species occupancy model that accounts for imperfect detection of each species while we're comparing our disturbances and stand ages. And because we fit this within a Bayesian framework, we're able to pull out essentially occupancy probability for each species at each iteration of this model and calculate species richness and other community metrics and the uncertainty around those. So diving into results a little bit here. One very clear trend that we're seeing is that bird richness increases with stand age in our plantations, pretty much doubling between 2 and 20 years after harvest. This isn't super surprising given there's really only one way to go from the start of these really low diversity plantations at 2 years, and that's up. In contrast, we see relatively high diversity immediately post-fire, but that stays fairly stable through time, such that the number of species supported by each disturbance kind of converges somewhere in this 10 to 20 year window. Mostly likely due to that immediate resprouting of hardwoods and shrubs and those really fresh snags immediately after fire. When we overlay salvage logging on top of this, we somewhat surprisingly see that salvage log stands support the same number of bird species as unlogged burns of the same age. And so I think all of this raises the question of, well, you know, this is the number of species that we're seeing supported. How do these relate to patterns of composition? And so for now, we've been looking at cavity nesting species and leaf cleaners. And what we see in terms of cavity nesting species is that they pretty quickly decline over time after fire, roughly halving the number of species between 2 and 20 years. And we see kind of the opposite trend after in plantations with a sort of a doubling of cavity nesting species, such that they again converge in these older stands. Leaf cleaning insectivores increase over time and after any disturbance, but especially sharply after the clear cuts because there's so little vegetation in those youngest stands. But once those planted conifers take off, they really grow quickly, whereas there's a lot more vegetation immediately after fire. And interestingly here, we haven't seen much difference between the unlogged and the logged stand, post-fire stands in terms of leaf cleaner richness. Then we have this third panel, which mostly is an area for me to further refine. But I think it's interesting to point out here that there are so many other species that we're seeing in the salvage log stands, and that seems to be compensating for the lack of cavity nesters and salvage stands in terms of our overall species richness. And so as we were kind of expecting in terms of this convergence, it seems like our assemblages in older burns are pretty well represented by plantation early cereal. We see similar diversity, both of leaf cleaning insectivores and of cavity nesters, and just overall bird diversity is similar in those older stands, regardless of disturbance. But these most recent burns are relatively unique. Just they have overall really high diversity, and the plantations don't seem to catch up to that until about 10 years at the very earliest. This contrast is particularly stark in terms of cavity nesters. Those aren't really as well supported by salvage logging either. And leaf cleaning diversity seems to be pretty well supported by salvage logging, but not really in these plantations at the younger stand ages. So my big caveat here is that any space for time substitution is imperfect, and we've put in permanent plots here. So my sort of long-term pipe dream would be to revisit some of these and get longitudinal data, but it's a pretty good chronosequence for what we have. And with that, I'd like to thank mostly all the field technicians who have helped me collect this data over the years. This is a pretty hairy area to work in, so mostly I just really appreciate all of them. And with that, I can take any questions.
My name is Jessica Barton. Today I'll be telling you about local to continental scale small mammal population dynamics in North America. So we know that small mammals interact with their environment in a variety of ways. So they can be consumers, consuming anything from woody and herbaceous plant material to mast material to fungi. They can also be prey to a wide variety of predators, and these predators may depend on small mammal populations. And they can also be host to ticks and other pathogens in their environment. So when there's this big pulse of resources or mast material, and I'll define mast seeding in the next slide, in their environment, there can be a big pulse in small mammals as well. And this can kind of cascade through the trophic levels, where if there's an increase in small mammals, there may also be an increase in predators and maybe an increase in number of ticks in the environment. And this has far-reaching implications into things like human health, where if there's an increase in ticks in the environment, there may also be an increase in the number of Lyme disease cases in humans or other tick-borne diseases in humans. So mast seeding is a synchronous and highly variable production of seeds by a population of perennial plants. It occurs in many species throughout the world, but the most recognizable to everyone here is probably acorns with oaks and cones and conifer trees. And like I said, it has this cascading effect through trophic levels, and these effects can be seen directly in small mammal populations. Masting can change them demographically, so their population size may change. So if they have more food, they might be able to produce more babies and therefore have a higher number of their population. It can also change them in a physiological way, so their body condition may increase or improve. If they have a higher number of food items in the environment or a higher number of nutritious food items in the environment, their body condition may increase, and they may also be able to survive overwinter better. They also can change behaviorally, so this can be movement into an area of high masting, so they can have a locally high population number in the masting area. So with masting, we see what is called an ecological dipole. So this is a fairly new term. It was coined in 2020. It's just fluctuations in ecological responses of opposite polarity in populations that are separated by large geographical distances. So basically, if it's on a continent, at one side of the continent, things that are happening, population size, reproduction, it'll be happening on that side and it won't be happening on the other side. And these patterns tend to flip or switch throughout the years. So for example, if it was east and west, in the east, it could be high one year, low in the west, and then the next year you might have high in the west, low in the east. So we also see what are called climate dipoles, and climate dipoles have been used by climatologists for years to describe these continent-wide weather patterns in all over the world. And here you're just seeing the temperature difference from 2005 to 2004 in North America. And this climate dipole is temperature. It's from east to west. So you can see these really hot areas in the east and cooler areas in the west. And this is a temperature dipole that goes from east to west. And in general, climate dipoles influence ecological dipoles in North America. So with mast seeding, we also see this east-west split, or this east-west dipole, with high sites of masting in the east and low sites of masting in the west in 2006. And the same is true for boreal bird eruptions. It kind of has this east-west split here with high counts in this year in the east and then low counts in the west. So we know that there's ecological dipoles in boreal bird eruptions and in mast seeding in North America. But what about small mammal indices? So we know that small mammals can be directly changed and their population sizes can change from mast seeding. If there's a high pulse of masting in their environment, their populations may change. So we wanted to know whether seed-eating small mammal species have an ecological dipole in North America. And our prediction was that there is a small mammal ecological dipole that will be reflected in negative correlations and population synchrony at large distances. And to complete our objectives, we used NEON data. NEON is the National Ecological Observatory Network. It is a network of 81 sites. They collect standardized data at all their sites. It's 34 freshwater aquatic sites and 47 terrestrial. This is just kind of a map of all their sites, all the little dots on their sites, and they have it partitioned into different areas in the United States. And they collect a wide variety of data. They have weather data collected at towers at each of their sites. They do tick drags, small mammal trapping, which is what we used, and they also collect plant productivity data. They have over 150 open access data products free for anyone to use and download from their website. So from all those sites, we picked 18 terrestrial forested sites because we were interested in masting and how masting would affect small mammals at those sites. And the data spans from 2013 to 2021. And then we also narrowed it down to have a more robust time series of data at each of the sites. So we only wanted sites that had greater than or equal to 4 years of data, and then also greater than or equal to 4 months of trapping within a year. And so this is this map of all of our sites that we're going to use for this study, or that we used for this study. And we also narrowed it down a little bit more. We only wanted to use seed-eating species. So from all the mammals trapped at those neon sites, we narrowed it down to 7 seed-eating species to determine their seed diet. We looked at Elton Traits 2.0, which just lists some functional traits of small mammals. So we used a diet of greater than or equal to 20% seeds. We also cross-referenced this with field guides and peer-reviewed studies to determine what these species ate. And so with all that, narrowing it down and stuff, this is what we get with our time series. So this is, every line is going to be a species site combination, and you're looking at the standardized count. So it ranges from 0 to 100. And I also want to point out this color ramp here. It's going to be used in some future slides and maps. Just keep in mind that 100 is very high. It's red, and then 0 is low and blue. So it's just a standardized count, meaning that the highest value at that site for that species is going to be set to 100. And the lowest value for that species at a site is going to be set to 0. So this is just kind of a way to compare them without having them on very, very different scales. So there's a lot going on in this, but I want to kind of point out these patterns right here. So in 2019, you can see these black lines are very high, they're at 100, and some of these grayer lines are here at 0. And then the next year, those same gray lines kind of get up to 100, and then those same black lines dip down to 0. So we can see that there is something going on. There's some asynchrony or synchrony. So these lines are grouped together, but they're kind of acting differently from these lines. But we didn't know where these sites were yet. So that's why we did a spatial synchrony analysis. We wanted to determine if sites close together and far away are synchronous or not. So we used a Correlogram analysis to do this. And this graph is basically going to have mammal count correlation on the y-axis here. So it ranges from -1 to 1. And then distance between sites in kilometers on the x-axis. So basically what you're going to see is a bunch of little points in the background here, and each point is a site pair combination. So of where it is going to be determined by their distance and their correlation to each other. So what we see here is kind of what you would expect. Sites close together are very highly correlated, and that correlation declines with distance. That's the Moran effect. But what we don't kind of usually see is this negative synchrony at sites that are above 2,000 kilometers or very far apart from each other. And that's what's sort of typical in an ecological dipole, where you see this asynchrony, this negative synchrony at sites that are very far apart. And then also I forgot to mention, if it's filled, it's significant. If it's not filled, it's not significant on this graph. And then just to sort of visualize this further, I have the same color ramp to remind you 100 is red, 0 is blue. I have these maps that I'm going to show you where the dots are going to also correspond to the values here. And so we don't use 2013 to look at the maps because there's only a few sites there. It was only in the east, and Neon hadn't gotten online yet, but this is 2014. I'm not going to explain every single map, but I'm going to point out some patterns in these maps. So here you can kind of see this grouping in the middle has higher values, while on either side of it has these lower values. And then in 2016, you kind of see that same thing. There's some higher values in the east here, but the higher values are also in the middle, and they kind of appear in California as well. In 2018, that pattern kind of flips, whereas before you saw some higher values in the middle, now there's lower values in the middle with higher values on either side. And then in 2019, you see this larger grouping here in the middle. It's a bigger grouping. It's not very small like it was before, but again, you see this kind of difference on the West Coast here. And nothing much is going on in 2020 or 2021. That's kind of typical of ecological dipoles, where in certain years you'll see this really strong dipole, but in other years you may not see strong patterns. So our prediction of this study was that there's a small mammal ecological dipole that will be reflected in negative correlations of population synchrony at large distances. So we did see that with our correlogram. We saw that between 2,000 and 3,000 kilometers, there was this significant asynchrony or negative synchrony in populations at large distances. But we don't know yet. We do see some sort of patterns in those maps where you kind of have this middle and possibly these outer areas that are different from each other, but we're not sure yet where it regionalizes out into. If it's a north-south, east-west dipole, we still have to do that work. So our future work is to regionalize the neon small mammal data. We want to also relate the small mammal data to climate data, such as precipitation and temperature. And then we also want to relate it and correlate it to mast seeding as well. And we're also doing some work in our lab to reveal tick and bird dipoles, also using neon data in North America. And what's interesting about this study is that most small mammal population studies are done at a regional or local scale, and to our knowledge, this is sort of the first time that we're kind of seeing these mammal patterns on a really large continental scale. And this work may be used in the future as a framework to discover other dipoles in North America and all over the world. And ecological dipoles might also be able to be used to predict wildlife dynamics at large scales if we see a really strong dipole pattern. And I would like to acknowledge my lab, the LaMontagne Lab at DePaul University, NEON staff and their data, and then the National Science Foundation for funding. And with that, I will take any questions.
So thanks, team, for the introduction. It's a pleasure to present on this session. So I'm Jaime Pinzon. I'm a research scientist with Natural Resources Canada. And I'm going to shift gears a little bit here with respect to taxon. So now I'm talking about spiders and ecosystem, so peatlands. Boreal wetlands in general are quite important ecosystems across Canada, and I'm going to be focusing mostly on Alberta, but they are very, very important resources of water, and are quite unique ecosystems. They provide very specific habitat for a great variety of vegetation and wildlife. And here in Alberta, about 20% of the surface is covered by wetlands of different types, as you can see here on the map on the right. Therefore, they are quite important ecosystem across the province. However, these areas are very fragile by human disturbances in general, and in particularly those associated to resource extraction like oil and gas. Most of these areas, timber is not merchantable, so these are not harvested, but many of these areas are highly affected by resource extraction. There are very large areas of oil sands within the province, and most of these areas overlap with many of these wetland areas here in Alberta. For those who are not very familiar about how exploration occurs in Alberta and in other parts of Canada and the world, I guess, most of this exploration is being carried out throughout seismic lines, which are relatively narrow corridors that dissect the forest. So these are they range from between 1.5 meters in the newer seismic lines to quite wide, about 15 meters wide for those that were created back in the 40s, 50s. These basically are used to identify and map the bitumen deposits in the underground. And therefore, that's why the forest is removed. So the machinery and crews can access these remote areas and are able to map the deposits. However, the main problem is that, as you can see on that map on the right, this has become a very pervasive disturbance across the province, and in there are areas where you can find seismic lines every 50 meters. So this is creating a very large impact across the landscape. And the main problem with this is that most of these lines, especially in peatlands, don't recover naturally. So they remain as the picture that you see there. This picture particularly was of a seismic line that was opened back in 1960s, and the picture was taken a few years ago. So you see that the line has remained intact after several decades of, following construction. So most of these lines are early successional, and regeneration in many of these is absent or very slow. So this has a very important environmental issue with respect to biodiversity and forest connectivity and so on. So one of the problems with seismic lines is ground compaction. We have been studying various peatlands across the province, and we have seen that most of these seismic lines, the ground is lower than the adjacent peatland, and this is basically due to the machinery that goes into these areas to open the forest, and then the machinery that goes afterwards for doing the exploration. So here, just showing some results from one of those peatlands where you can see the relative elevation between the seismic line in orange and the adjacent peatland in green. And you can see that consistently, these lines are much lower than the adjacent forest. And so we have been measuring bulk density in these areas just to have an idea of soil compaction, and it's quite evident that the soils are highly compacted compared to the adjacent forest. And even though these lines are opened and used mostly during the winter, this doesn't, and the idea for this is kind of like reducing soil compaction. It seems that at least in peatlands, this doesn't solve many of these problems. So with soil compaction, there are other problems associated to these, which is higher moisture. So this is a different peatland where we have been tracking soil moisture since the spring of 2019 until this date. And that green line is the soil moisture on seismic line, where the green line, the gray line is the soil moisture in seismic line, and the green line is the soil moisture in the adjacent forest. The blue line is just precipitation. And you can see that consistently, the seismic line, the ground of the seismic line is much wetter than the adjacent forest. And of course, this has very important implications for the whole hydrology of the peatland, regeneration of trees, and establishment and survival of these tree species. And overall, we're expecting that these will have some impacts on biodiversity. So the idea of this ongoing project is not only document the composition of spider assemblages in Alberta peatlands, which are very poorly understood and known, but also kind of like have a contribution to the current biodiversity knowledge of spiders in the province, and tag along with evaluating these spider assemblages and trying to understand how they are responding to the fragmentation associated to these linear disturbances that I was talking about before. So for this study, we have a number of peatlands across the province. Many of these are located, agglomerated within that Fort McMurray area. However, we have a few other peatlands outside of that area. And this year we are collecting in a different peatland. And the idea is that over the years, we plan to add more peatlands to have a much better idea of what is going on in these areas. For this, we collected ground-dwelling spiders using pitfall traps throughout the frost-free season, which is normally May to August. And we have been collecting data for the last 5 years. So these different sites have been collected at different times, but the one commonality is that all these areas have been disturbed by the presence of a seismic line. So when we think about, when we look about overall species richness, overall we have observed almost 300 species of spiders in these different peatlands with over 55,000 individuals. So one of the things that had been quite surprising is that apparently spiders really love peatlands. They are very abundant in peatlands compared to uplands, which they are also quite abundant, but they are also quite diverse. And one of the very, I guess, enjoyable part of this work has been being able to report a number of species that were previously not known to Alberta. And this is basically because peatlands are normally poorly studied, and people basically don't like to get wet, so they don't access these areas. And up to now, we have a very limited knowledge of the species composition. So here is the species richness across the different sites, with an average of 87 species across the different peatlands, but you can see that there's quite a bit of variation among them. Now, when we look at the species richness by site and habitat, so here is comparing the adjacent forest, which is the dark gray, with the seismic line, which is the light gray. There's a lot of variability as well across the different peatlands, but we find some, we see some consistent patterns. There are some of these peatlands where the seismic lines have a much greater richness, whereas there are others in which the richness in the seismic line and the adjacent forest are more or less similar. There's only one peatland among these 18 where the adjacent forest had a greater species richness than the seismic line. So kind of like to summarize all these data, consistently, these seismic lines, which are very open areas, are attracting open habitat species, and they are, of course, influencing the overall species richness that we might see across these landscapes compared to the undisturbed adjacent forest. So when we look at the composition across the different sites, of course, as we were expecting, there are important differences in what species you find depending on where you are in the province. And there's quite a marked difference in species composition between the Grand Prairie area, which is our northwestern peatland, compared to the Brazeau area, which is kind of like a southwestern peatland, and all the other peatlands in the northeastern part of the province. So there's a significant compositional change across the province, which again was expected, but anyways, quite interesting. So after accounting for this site-to-site variation, we started asking, okay, what happens when you are comparing the seismic line and the adjacent forest with respect to composition? And it's very clear that the composition on seismic lines is very different from that in the adjacent forest. There are a number of species that are shared, but those seismic lines are mostly dominated by open habitat species. And this pattern seems to be driven by soil moisture, which is that green arrow that you see there, volumetric water content. So as I mentioned before, these seismic lines are much wetter, and these conditions seem to be driving the composition on these very open areas. So there are a few take-home messages that I want to bring from this research. And of course, one is the importance of peatlands for ground-dwelling spiders. They are quite important, but there's still a lot that we don't know, right? There are many records for the province that they are coming out from this research, and most likely there will be more as we explore other peatlands. It's very interesting that even though we have very low number of localities, the total number of species that we've recorded across these peatlands is about half of the species recorded are known to the province. So these are very important habitats for spiders, which has very important implications for management as well, given that difference in composition across the province. Interestingly, we found greater richness along those linear disturbances, and as well different species compositions related to that adjacent peatland. So this has a very interesting problem that creates a very interesting problem when we think about forest management and oil and gas exploration and all the implication that comes from restoration that we are just starting to understand. And this is ongoing research, so there are more peatlands being assessed, and hopefully within the next couple of years we'll have a much clearer understanding of what is the effect of these linear disturbances on spider biodiversity. So thank you very much for your attention, and we have many people involved in these different projects, so I just don't list them here, but thank to all those that were part of these. And I will be more than happy to answer any questions if there is any time left for doing so.
Well, first off, I'd like to say that this work isn't really all just me at all. It's actually a collaboration between two very productive people. One is Lisa Veneer, who actually came and organized the conference for us, and also with Dave Morris, who's also a co-organizer of the conference. And what I'm going to do today is pretty much give you kind of a follow-up situation of a study that we started about 7 years ago, looking at the impacts of biomass removal in jack pine stands in Ontario, essentially. Well, let's go over here. There we go. Well, biomass harvesting has kind of been on and off since the 70s, where we've kind of been into it and out of it, and now we're really back into it. And I think you see this a lot in Europe, particularly where a lot of countries have pledged to kind of kick fossil carbon in general and go to completely sustainable sources of energy. And for northern countries, particularly like in Scandinavia, this has meant moving to heavy use on wood chips and biomass that comes from basically often non-merchantable sources of timber, like stumps and branch wood and things like that. And more Canada has been thinking about it as well. And this has become part of Ontario's energy plan for sustainability, part of Quebec's plan for sustainability. And with the war in Ukraine and all the relationships with oil producing companies, and I'm sure that biomass energy is taking on a new kind of importance. And we've seen this is, there's a recent article in Science, kind of a perspective piece talking about, in Nature, talking about how the war in Ukraine is actually going to maybe one avenue by which countries may go further into biomass energy production. So what I'm going to talk about is in large part, I'm going to talk about the Island Lake Biomass Harvest Project. And this is a study that is kind of near Chapleau, Ontario. It's not too far from where the conference would have been in Sault Ste. Marie. But the goal of this project was basically to put on a very experimentally rigorous test of what would it look like if we were to take non-merchantable wood out of managed stands for increased production of biomass and woody biomass in terms of chips. Let me direct your attention down to this kind of map with the blocks here. What this study was an experimental study where we basically had 4 different levels of biomass removal. We had kind of a standard tree length harvest, which would be basically like as if you cut the tree and left branches and a lot of the non-merchantable tops on site. we did what we would call a full tree harvest, which would be where the tree and all the branches was moved to a landing and delimbed at the landing. So it's an export of not only the trunk, but also the branches. Then we started to do some kind of intensive Scandinavian type things. And one would be the removal of stumps. And this was done with kind of a stump plow to remove the stumps from experimental plots. And then we did something that nobody would ever really do. And this was blading of sites. And this was essentially where all woody biomass and the organic layer was stripped off with a bulldozer. And this wasn't really meant to be like a actual option that people are thinking about doing, but it was meant as a rigorous experimental control to be kind of at one end of this biomass, the extreme end of this biomass removal. Now, over here to the right, you can see what these treatments look like in terms of the volume of dead wood. But today what I want to talk to you about is kind of the additional things that go on with these biomass removals. Anytime we do kind of a silvicultural treatment, you have to realize that there's more to it than just the actual treatment of removing wood. You have things like soil compaction, machineries going in. And another thing that's really important, particularly here, is the applications of glyphosate, because one of the things that you want to do for this to be sustainable is you want to have the trees come back. So what happens is you go in, you take the biomass out, and then you usually have 3 or 4 or 5 applications of glyphosate to make sure that the next generation of trees reestablishes. And I thought this was kind of a nice full circle approach to the speaker that we had this morning, or the keynote we had this morning, where we're back talking about potential impacts of glyphosate. If you're really interested in the initial impacts of what happened with the biomass removal, Lisa has already published this in a really nice article that's in Canadian Journal of Forest Research, and I put the reference for it down here. But what I'm going to talk about is the 7-year post-harvest response to see if, one, do we see any recovery coming back from in these biomass plots, and 2, what is the additional impacts of glyphosate on arthropod communities? So let's go over there. So just to give you an impression of what these treatments look like right after we did them, this is kind of, we're going to go kind of clockwise starting in the left-hand corner here, but this is what a normal tree length clear cut would look like. And this is kind of standard practice that we would do in Quebec in terms of dead wood volume. If we move over to the right, we see what a full tree harvest looks like. And this would be kind of standard practice, what you would see in Ontario generally. And then, sorry, we're not actually going clockwise, but if we go diagonal over to the bottom left here, we see the stump harvesting. And you can see that starting to get pretty bare in terms of, you look along these kind of corridors here, and you see that there's not very much dead wood anymore. And then if you go to the blading treatments, it's essentially a sandbox with very, very little vegetation, no dead wood at all. But things change. 7 years out, things are starting to green up there. If we look at the tree length, this is just kind of the same series of photos, but with the vegetation that's coming back. One of the things I wanted to show you is that there is a fair bit of vegetation coming back in this tree, in both the tree length and the full tree, and even the stump harvesting. But the blading sites are still relatively open, and there is really not much other vegetation there to speak of other than the trees that were planted. Lisa sent, this is a photo that was taken by Laura, but that was sent to me by Lisa, but this gives you a really good impression to look at, like, what does the glyphosate treatments look like? On the right, we have a full tree harvest plot with glyphosate. On the left, we have a full tree harvest plot without that was not treated with glyphosate. And you can see there's quite a difference in terms of the vegetation. Now, so we took this island lake experiment where we just show, and we wanted to compare this. Where do these treatments kind of fall along on the range of natural variability that we would see in this area. And so we have 3 reference sites that we use in association with these sites. And one is a young stand that was a, sorry, I got to zoom things in here. It's a young stand, 56 years old, and this came from a clear-cut origin. So it was a stand, and this is actually in the 2nd rotation here. We have an older stand that was from fire origin that's 97 years old, and this stand is starting to break up quite a bit. There's a lot of dead wood that's down there, but these 2 areas, they have a fair bit of overstory cover, and there's trees in the understory that are providing shade. There's vegetation cover essentially. In contrast, one of the ones we had was one of our last reference studies is a recent burn. And this is a burn that happened 8 years ago. It was a plantation, but it burned quite heavily. It was a heavy crown fire. And so while there's a lot of wood here, there's not very much overstory at all. It doesn't look anything like these other 2 plots. This is some data that Dave Morris gave us. One of the things that you want to see, this is actually what we're looking at here is comparison of shrub and herbaceous cover in the island lake biomass sites, which is to the left of the graph, and in the reference sites. Now, in the reference sites, we never applied any glyphosate. And it's important to point out that we did apply glyphosate in the island lake sites, and it's exactly like what you would think is there's no vegetation coming, or there's no shrub cover or vegetation cover coming back with that glyphosate. So you can, in terms of what these reference plots kind of look like, in terms of percent cover, the biomass plots, particularly some of these ones with less intensive treatments, there's a reasonable amount of vegetation cover that's come back, and that seems to be kind of similar to what we're seeing in the burn. Okay, so what we did was in all of these plots, we basically set up kind of a series of pitfall trapping, and most of our plots kind of look like this, where we have a large square plot, half of it's replanted with jack pine, half of it's replanted with black spruce, and then if you draw a perpendicular line to that, half of that's treated with glyphosate, half of it is left alone. Within these plots, we put two sets of pitfall traps within each of these subplots. And what we did was we monitored ground beetles, rove beetles, and spiders in the exact same way that we did from the initial part of the study. We identified all this material to species. Actually, there are some people involved here. I think Ursula was one of the responsible who helped identify the carabids. The rove beetles were verified by Jan Klimaszewski, and I believe the spiders were verified by Joe Bowden. And I think Lisa can correct me if I made a mistake with that. What I'm going to show you now is kind of the community level responses in much the same way that Lauren showed you at the beginning of our session. So when we look at ground beetles, the first thing I want to do, I want to kind of start at the bottom of the tree, and I want you to look at these kind of endpoints here, because what these are distinctive assemblages of organisms that are based on the composition, the abundance, and the species richness of the animals that are there. And the first point I want to make is that all 3 reference stands come out having distinct compositions in terms of ground beetles. Whoops, I think I have a typo in here. All the ground beetles are, oh God, darn it, there we go. The ground beetle composition differs amongst all the 3 reference stands. And an important thing is that these are all very different from everything else we see in the biomass plots. So this is what this is telling us is the biomass plots are essentially falling outside the range of natural or the range of the reference conditions that we're seeing. And we're not seeing much recovery in terms of the ground beetles in 7 years. The next thing I want to point out is the burn sites are, or the blade sites are very different from the rest of the biomass treatment sites, and that's not too difficult to be expected. You saw what those things look like. It's a sandbox. But one of the things that's interesting is when we look at the rest of the biomass plots, in plots where we did not apply glyphosate, we don't really see any differences between the tree length and the full tree treatments. Those are basically kind of looking like the same in terms of assemblage. But what happens is when we apply glyphosate, we do see that this starts to exacerbate the differences between the full tree and the tree length sites. And essentially what it's doing is it's starting to pull this glyphosate, this full tree plot is starting to look more like the stump harvest. When we go to the rove beetles now, rove beetles is a little bit easier, a little bit more simpler. First of all, again, we see that we have we have three different, the reference sites are all different in terms of composition, but unlike ground beetles, now the burn sites are more similar to the biomass removal sites. When we look at the response in the biomass removal sites, blading again is its own thing, and we don't see any differences between the biomass removal for rove beetles. And finally, there's no effect of glyphosate in these plots at all. Now, when we look at spiders, again, we see three different responses in the reference things. Burns are again closer to the biomass plots. Blading, again, is its own thing. But unlike the ground beetles, when we add glyphosate, what we do is we kind of homogenize the whole plot. And so basically what we don't, when we add glyphosate, we can't really see any difference between full tree, tree length, and stump plots. And in fact, when we actually look at the composition in terms of all the species signatures with this, they tend to look a lot more like the stumped treatments that we see in without glyphosate. And so essentially what is happening here is the glyphosate is kind of slowing responses. It's making everything look like a more opened, more disturbed stand, and it tends to be slowing recovery of spiders essentially. So just a couple take-home messages. First of all, we're not really seeing any recovery yet, and that's not too shocking. It's only 7 years out. We probably are going to need, if you remember back to Lauren's presentation this earlier in the session, she hasn't seen any recovery yet or she didn't see recovery in the biomass plots in Quebec in ground beetles for about 15 years. So it's probably pretty early to expect recovery. But what seems to be going on right now is a lot of these biomass plots seem to be falling kind of outside the range of the variation that we see. And if there is any kind of analog that's close, it's probably closest to these burns. And that is probably related to, one, there's no overstory in the burns really to speak of anymore. And the vegetation seems to be coming back into that because we haven't, the vegetation seems to be coming back, particularly in the non-glyphosate plots. But when we do add glyphosate to these sites, we seem to be kind of compounding or prolonging the effect of the biomass removal. So this is something that's probably going to slow recovery in these sites, but it could be one of these really easy fixes where we just don't spray so much glyphosate and maybe we could speed recovery. So I can see that I'm already hitting the time limit. So what I'm going to do is just drop it there and ask, do we have any questions?